This is a lecture to tell you about the basics of QTL mapping. So this is Roman numeral 2 in our topic of quantitative traits. Roman numeral 1 was a reminder that many traits are quantitative traits, meaning they are influenced by a variety of alleles, more than one allele, and are also influenced by the environment, right? So when we have our if we look at any individual, even if they have the same genotype, we're going to see a histogram of types of traits. When we make F1s, we're going to see, again, variation in the trait. And among our F2s, we'll have even more variation. So that's a hallmark of quantitative traits, traits determined by multiple loci. All right, so QTL mapping is one way to actually identify the places in the genome that are responsible for the differences among individuals. All right, so we're going to look at two methods, and one of them for this video is QTL mapping. So QTL, quantitative trait locus or loci. So one thing we need for QTL mapping is marker loci, and these are places in the genome where we know the sequence, and we know that the sequence in one part of the genome differs from a sequence in another part of the genome. So it's really literally a map. Like the marker loci are the ways that we know where we're talking about in the genome. Okay, so these aren't necessarily the loci that are influencing the traits. These are just sequences of DNA that we know where they are in the genome. And I think this will make more sense as we get into the logic of QTL analysis. So we're going to use these marker loci to essentially look along the length of the DNA molecules, along the chromosomes, and identify places where we find genes that aren't the marker loci, but other genes that influence our traits. Okay, so that's the logic. We're going to introduce ourselves to the system a little bit that we're using to, as an example of this quantitative trait locus mapping. So this is a system of flowering plants called monkey flowers, is the genus Mimulus cardinalis and Mimulus lewisii. And there they are. They're sister species. They have overlapping geographic ranges, but they are pollinated by different species and they look really different. If you bring them into the lab, so A and B here, top left and top right, are Mimulus lewisii and Mimulus cardinalis. So that's what they look like. And if you take them into the lab and hand pollinate them, you get F1s that have intermediates. This is the lovely, classic codominant sort of phenotype, right? Okay, so F1s look intermediate. In nature, we don't see any F1s. So that's a question. Why we don't see any hybrids? Why are there no hybrids in nature? This suggests that the specific traits of these two species are adaptive for some reason, and the traits of a hybrid are maladaptive, right? So hybridization is not favored. Selection is acted separately on these two species to push them closer towards their phenotypes that are adaptive in whatever contexts they are. Okay. We can also cross F1s, and when we cross F1s, we get wide variation among our F2s. What does this suggest? Think for a minute. Okay, variation among our F2s suggests a couple of things. One, that the phenotypic differences are determined by underlying genetic differences. And specifically, we are looking at a case with multiple genes involved, a quantitative trait. Okay, if this was just one gene, our simple Mendelian model, in our F2s, we would have our 1 to 2 to 1 ratio. Okay. Oh, right, I already said this, hybrids not found in the field. Why not? What about these flowers might lead there to be little opportunity for hybridization? You're thinking. Maybe the traits have something to do with pollinator attraction and the type of pollinator that's attracted to a Mimulus lewisii flower is not going to go to a Mimulus cardinalis flower and vice versa. So there's no opportunity for hybridization. So it's not that the flowers are like thinking about not hybridizing or that they're like trying to not make hybrids. Maybe occasionally a bumblebee lands on the wrong flower and sends some pollen. So maybe occasionally we make hybrids, but they don't make offspring because no pollinator is attracted to this intermediate sort of type of flower, this blue flower. Right? So maybe the traits that have been selected upon to lead to these different phenotypes have to do with pollinator attraction. We're going to come back around to that once we talk about how to do QTL mapping. Okay. Okay, so we know from phylogenetic analyses that the most recent common ancestor to these two sister species was likely bee pollinated. So then we're looking specifically for traits that lead to or that cause a transition to hummingbird pollination in this lineage, Mimulus cardinalis. So that's our question. What we want to know, what parts of the genome are associated with these phenotypic differences between lewisii and cardinalis. So they've got lots of similarities. There are genes responsible for that. We want to know what are the genes that are responsible for the differences. How do we figure that out? Right? We can't just look at the genome and like see what the genes are doing. Right? We need to do a bunch of crosses. We need our marker loci, and we need to look at phenotypes of individuals with known genotypes. We also, because this is a quantitative trait, we're going to use our understanding of linkage disequilibrium to get at this question. Okay, so here's the outline of the method. Then we're going to look really closely at some of the underlying logic and then look at the results. So the method. We've identified our marker loci, our little flags that say, if we get this sequence, we know we're talking about this little part of the genome. Right? So we have these across the whole genome. We know which marker loci correspond to which parts of the genome. We also need individuals, or we need we need to choose marker loci for which the species is homozygous. So there's only one allele for this particular marker locus in the species. Okay. So Mimulus lewisii, it's going to have this place in the genome. It's got a different allele. Every individual of Mimulus lewisii has that allele. Mimulus cardinalis is also going to have this place in the genome, but a different allele. So if we just looked at the sequence for the marker locus, we would know what species that little part of the genome came from. Okay, then we're going to go in our greenhouse and we're going to cross lewisii and cardinalis individuals and generate hybrids, F1 hybrids. And these hybrids are going to be heterozygous for all of those marker loci. They're going to have one cardinalis allele. They're going to have one lewisii allele. Okay, then we're going to take our F2 or F1s, mate them together, and get an array of hybrids, F2 hybrids. Okay, so they're going to look like the rest of the pictures in that box. They're going to have differing phenotypes because of their differing genotypes. Okay, and then we're going to have some way to score the phenotype. So we're assessing the phenotypes. If it's pigment, maybe we're extracting the pigment and determining the quantity, or maybe we're comparing it to some color scale that we've set up. Maybe we are measuring like pollinator visitation, and so we're going out in the field and watching, or we're introducing pollinators somehow, and we're watching which particular flowers the pollinators tend to go to. Okay, maybe we're looking at the quantity of nectar, and so we're extracting the nectar and measuring the quantity. Maybe we're measuring the amount of sucrose in the nectar, so we're doing some chemical analyses, right? So whatever traits we're looking at, we need some way to measure those traits. Okay, so that's what we do. Why are we doing this? What's the logic behind this? Oh, sorry. We also have to determine the genotypes of each of the F2 individuals at the marker loci. So we just do this with basic genetic analysis sequencing. Okay, figure out what genotypes the individuals have at their marker loci. So then we can make a connection between phenotype and genotype. Genotype for the marker locus, which might not be actually controlling the phenotype. Okay. So stepping back from that example, let's talk a little bit about the logic behind QTL mapping. Okay, so what this figure is showing you, we're looking at the genome for one marker locus for one individual, one diploid individual. So here, so M is for marker, Q is for QTL, potential QTL, quantitative trait locus, and the subscript refers to the species. So here's our marker we've identified for loci, the allele that's only in loci, the marker allele that's only in cardinalis, and maybe this is an example, maybe we've got a trait, or sorry, excuse me, a gene here that controls the phenotypes that we're interested in. Okay, and so we're going to have a gene, an allele form that's specific to loci, and our gene with its allele form that's specific to cardinalis. I'm wondering if you're seeing my cursor. Now I have a laser pointer. I know you're seeing this. Okay, so our marker locus, potential phenotype influencing gene with loci alleles, potentially phenotype influencing gene with cardinalis alleles. And the line is trying to indicate physical linkage. So the marker locus and the QTL are close to each other, physically close to each other. So we expect them to be inherited together. So we make our F1s. We cross these two individuals, the parents. We get F1s. They're all hybrids. They have a mix of haplotypes. Okay, now we've got 4 different alleles here, but only 2 possible combinations. 2 possible haplotypes. We've got blue with black and pink with yellow. We don't have blue with yellow or pink with black because these loci, the marker and the quantitative trait locus, are linked. They're physically linked. So these are in linkage disequilibrium. I'm pausing so you can think about linkage disequilibrium. Maybe you're pausing yourself and looking back in your notes. What does that mean again? All right, so we're going to cross our F1s and get F2s. This is just our process. Now, if these loci, the marker locus and the quantitative trait locus, if they are linked physically, we are only going to get 4 possible, 3 possible offspring F2 genotypes. So we're going to have individuals who are homozygous black and homozygous blue, individuals who are homozygous pink and homozygous yellow, and individuals who are heterozygous for both in this specific way, pink and yellow together and blue and black together. If these QTL influence the trait that we're looking at, when we measure the phenotypes, we're also going to see clearly delineated differences in the phenotypes among these 3 genotypes. So we're going to have individuals who are this phenotype distribution, so low in whatever trait we're measuring, intermediate for the heterozygotes, and high for the others. Right, so what do we know about these individuals? We know that they all share being homozygous for this blue marker locus. We don't yet know, QTL analysis doesn't tell us what this possible black gene is. Okay, it just tells us that individuals who are homozygous for this, the loci marker locus, share phenotypes in common with each other. individuals who are heterozygous have intermediate phenotypes, and individuals who are homozygous for the other species marker locus share a phenotype similar to each other that's different from the phenotype caused by the other marker locus. So this doesn't tell us anything really about the quantitative trait locus that's in charge, but it tells us that there's potentially a quantitative trait locus near our marker locus. Okay, let's look at an alternative explanation, or alternative expectation rather. So in this case, we've still got our labeling color scheme, but we don't have the lines indicating haplotype. So here we're saying that blue and black, pink and yellow are not linked to each other. So we're going to expect crossing over, independent assortment among marker loci and quantitative trait loci. And so then you see that our F1s are just like our F1s from before, because that's how meiosis and fertilization works. But then our F2s have all the possible variety. You've got some that are homozygous for the loci marker locus, blue, and then among those, you've got 4 different possibilities for the quantitative trait locus. For your heterozygotes, heterozygous for the marker loci, you have all 4 possibilities for the quantitative trait locus. And for individuals who are homozygous for the the cardinalis marker locus, again, all 4 of the possible combinations for the quantitative trait locus. So among your whole bunch of F2s, you're going to have this wide variety of phenotypes. So in this case, our marker locus doesn't indicate anything about the phenotype. There's no relationship between what genotype you have for the marker locus and what phenotype you have. Okay, so this tells us that this marker locus, this flag in the genome, this particular flag is not near a gene that is contributing to differences among these 2 species. Right, so we're using lots and lots of marker loci. For some of them, we're going to see the pattern like at the bottom. There's no gene really close to this marker locus that's influencing the phenotypes we're looking at. Some of them we're going to see like the scenario at the top. There's a strong association between where this marker locus is in the genome and what phenotype you have. And we might have some in between, right? Okay, so that's the principle that we're using. So we've got our marker loci. We are using them to scan the genome, looking for correlations between where the marker locus is and what phenotypes the individuals have. Okay, so back to the study, they looked at 12 traits related to pollination and 66 marker loci across the genome. I've already told you, what does this above distribution indicate? I'll give you a second to think, remind yourself, what does this distribution indicate? This means that there is a correlation between what genotype you have for the marker locus and what phenotype you have. So that tells us that there is potentially close to this marker locus a gene that is involved in determining this phenotype. All right, here are some results from this Bradshaw et al. Study. So what we have on the x-axis is the percentage of the variance in the trait that's explained by the QTL. And this is a histogram. On the y-axis, we've got the number of QTLs that have that amount of explained variation. And we're looking at the Mimulus lewisii genome and the Mimulus cardinalis genome. Okay, so what you can see is that most of the QTLs that they examined, of the 66, explain a relatively small amount of the variation. And there are a few loci that explain a lot of the variation. And the color coding are telling us what traits specifically these are related to. So are they related to attracting pollinators, providing a reward like copious pollen or lots of nectar, and efficiency of pollination. So does the flower structure like allow for a bee to land on it, for instance, or does it is it structured in a way that deposits the pollen right where it needs to be to transfer it to another flower? Okay, so most of the QTL explain a small amount of the variation in the trait, and a few of these QTL explain a lot of the variation. So then these we're thinking are the traits that are strongly influencing the differences between our two species, the traits, the genes specifically, that would be under strong selection in this system with our two separate pollinators. I said that. Okay, so then to confirm all of this, to connect our genetic results, our histograms about what the trait is, to actual natural selection reality, we would then want to put out our array of F2s of known genotype, known marker locus genotypes, and make observations. Which F2s are pollinators most likely to visit? And we've got two kinds of pollinators. So we've got hummingbirds and bumblebees, and we can watch them separately, and we might see that hummingbirds tend to favor certain traits and ignore other traits. Like maybe they don't care so much about the shape of the flower as the color of the flower, or maybe they don't care so much about the color of the flower as the amount of nectar that's in there. Maybe bumblebees care about different things. Maybe bumblebees are really targeted in on color and don't care so much about other things. Okay, so we do this. We we get our grad students to sit in the field and watch all of these F2s and record pollinator visitation. Like, do they land? How long do they sit there? That kind of thing. Maybe we're catching some pollinators and seeing how much pollen they have on them. Okay, so we're doing this study, and then we'll be able to link the genotype that an individual has with its likelihood of getting pollinated by a particular type of pollinator. And here's a little summary of some of the results that they found. So one thing that they found was that bees visit flowers that are more yellow less often than they visit pink flowers. So bees seem to be really keying in on the color and specifically preferring pink flowers. Hummingbirds seem to be keying in on the amount of nectar. So they visit flowers that have more nectar more often than flowers that have less nectar. Okay, so we're thinking these are two traits that are strongly influenced in the evolution of these two species. Okay, I'm actually going to skip this part. Okay, so that's great. We know a lot now. Now we know that we've got where we are in the genome. We know that near this place in the genome, near our marker locus, there's a gene that influences traits. And it doesn't just influence traits, it influences a trait that is useful in this whatever environmental context, that increases visitation by bees or increases visitation by hummingbirds. Okay, so this doesn't tell us everything. We still don't know what those genes are, and we don't know exactly what we don't know what they are, right? So of course we don't know what their protein products are. And that's where the second method, candidate locus investigation, comes in, and that's going to be in a separate video.